At this meeting, we've begun to hear about some other subsetting of lung cancer that gives us excitement about potentially some new therapies in the near future that can help patients that have not been helped before. One of these has been the discussion of a small subset of patients with what are called ROS1 rearranged lung cancers. This has been an interesting story. This was presented by Alice Shaw out of the Massachusetts General Hospital group, a fantastic group that's really helped show a lot of the excitement and advances in lung cancer over the last several years. What Alice showed is a discovery of a rearrangement that probably is in a small subset of patients with lung cancer that we call ROS1 rearranged lung cancers. This probably is in about 2% or so of patients with non-small cell lung cancer. And what's been interesting is this fits this clinical picture or phenotype that seems familiar. Patients who are predominantly non-smokers, patients with adenocarcinomas, seem to be the ones that potentially could have this rare rearrangement. Now what's interesting, and Alice presented about 14 patients who have been in a study who all shared this common genetic rearrangement, that is to say this ROS1 rearrangement. What's interesting is that this rearrangement in a tyrosine kinase shares identity with rearrangements in the ALK tyrosine kinase, such that the drug that's used in patients with the ALK rearranged tumor, in this case crizotinib or Xalkori, seems to be effective in this rare subset of patients with lung cancer that have ROS1 rearranged lung cancers. And so to say things more simply, a drug that's on the market as of August of 2011, Xalkori or crizotinib, appears to be an effective agent also for these patients with this other rare rearrangement. So Alice showed that patients with these 14 patients with ROS1 rearrangements so far appear to predominantly benefit from treatment with Xalkori or crizotinib. I believe her response rate was just under 60%. It's early, but it appears there are many patients who are now on this therapy out many, many months. What was most telling was an anecdote of one of the patients, a 31-year-old gentleman who had unfortunately progressed despite prior modern systemic therapies. A discovery was made that this patient had this ROS1 rearrangement. This patient was treated with crizotinib or Xalkori, and in about 8 weeks, 2 cycles, had a complete response, a complete disappearance in their cancer. And I think that one example gives a lot of hope that we may soon be able to have a new therapy for patients with this rare rearrangement. It wouldn't take a great leap to imagine if you could just find 20 or 30 more patients and demonstrate that same type of dramatic response and treatment benefit, Xalkori's label will expand and be used in this setting. So I think that's very exciting, and I think we all expect ROS1 testing to become more standard across the community. Switching gears here, despite all the excitement about trying to subset and better understand these molecular subtypes or subsets of lung cancer, we had a very exciting presentation. And in our group, Sarah Cannon had the privilege of being a part of this study. This was presented by Julie Brommer out of Johns Hopkins. This was a study looking at a different approach to treating lung cancer. This is not a selective approach where you're only talking about 1 to 2% of the population. This was using a therapy to help boost a patient's immune system, specifically their T-cell immunity to fight cancer. This idea of boosting immunity to fight solid tumors, in particular lung cancer, has not been a strategy that's had a lot of optimism in the lung cancer research community. So it was exciting to see these results. This was a phase 1 study looking at a novel agent from BMS that more simply is referred to as AntiPD1. This is an antibody that, an antibody-based therapy that targets what maybe is said simply as the brake to immunity. So by taking the brake off the immune system, you can activate T-cell immunity to hopefully fight cancer. What Julie showed is that this drug appears to be active in patients with very refractory lung cancer. So she had about she had a expanded cohort of patients who had received different levels of this anti-PD1 therapy, Now, across the board, if you look at who these patients were, they were mixed groups of patients. So these patients had squamous cancers, they had non-squamous cancers. These were patients who had received one prior therapy. Actually, most of the patients had received more than at least three prior therapies. And when you put all these patients together, you can see that there was about a response rate of about 18%. Now, that may not seem high, but when you think about a group of folks who've been heavily pretreated, most of these patients at least three priors, to see about 1 in 5 of these patients have shrinkage in the cancer was quite dramatic. And I've had, again, the privilege of treating some of these patients, and can think of 2 patients who, while not having a response, have had prolonged disease control. And that is quite rewarding to see, especially when you think about this agent not being very toxic. This agent in general was associated with fatigue and what I would call constitutional symptoms, but significant or severe toxicity was quite rare. And there were 2 patients in the overall subset who had some pulmonary toxicity that even contributed to death, but that was felt to be quite rare. And I think this is an exciting story. So the next step is to see how this anti-PD1 or immune-based stimulant compares to standard chemotherapy, and that's what's expected to be the next step in the evolution of this agent's life cycle. So hopefully, if that can beat chemotherapy head-to-head, this could be an improved drug in the treatment of lung cancer, and likely going to be a drug that's not limited to 1 to 2% of the population. In fact, could be a drug that's available to all patients, regardless of histology or other genotypic features. One trial that we've heard news about is an exciting study, and the lead presenter is Dr. Pasiani from Dana-Farber. This is an exciting trial that I think is going to get a lot of attention. I've heard some say, actually, this may be the most exciting thing coming out of ASCO this year in lung cancer. This is a randomized trial in patients with pretreated lung cancer, but what's different about this study is this is taking a group of patients who have something called a KRAS mutation. We know that KRAS mutations are pretty common in lung cancer, maybe anywhere from 1 in 5 to 1 in 3 of patients we see just across the board have this mutation, and this mutation has been one that's been associated with a poor prognosis. What's been very challenging is we haven't had any therapies that have shown benefit in this rather large group of patients with lung cancer. What Pasi is going to show us is that if you take these patients and randomize them to chemotherapy, in this case docetaxel or Taxotere, or that same chemotherapy plus an agent that's called a MEK inhibitor, a MEK inhibitor, and this is an oral agent from AstraZeneca, it's AZD6544, that patients actually have improvements in several parameters. So the trial was actually designed to look at a primary endpoint of overall survival. And what they showed in this trial, and the numbers are small, it's about 80 patients total. They had to screen several hundred to find 80 patients. But in this trial, with those small numbers, they had a positive trend in favor of survival for the MEK inhibitor with Taxotere versus Taxotere by itself. Also, the response rates were quite dramatic. As I recall, it was about no patients responding in the chemotherapy alone arm versus approximately 2 in 5 patients responding in the MEK inhibitor chemotherapy arm. And there was also a hint that progression-free survival, how long you live before your cancer gets worse or you die from any cause, that appears to be an advantage in favor of the combination. So I think this is pretty exciting. This is the first time we have a group of patients that's, again, not a 1–2% subset of patients with lung cancer, but perhaps maybe as high as 1 in 5, 2 in 5 of the patients we see every day that come in through the front door who potentially are benefiting in real ways. And it appears to be perhaps a benefit in terms of survival. And I think the next step is to do a larger trial to get over the issues maybe that prevented this from being statistically significant, to show that this combination is probably better than chemotherapy by itself. So I think, you know, this is just one of a few things that are very exciting as we move forward in this new era of molecularly targeting lung cancer that seems to have some legs underneath it.